We'll start the discussion this morning with Aubrey talking about raising the glass ceiling imposed by aging. That's interesting. I'm very interested in that. So let's hear from you, Aubrey.
Thanks very much. Yeah, good morning. Is that going to work? Let's wait until it does. There we go, marvellous. Yes, thank you very much for getting up in time. I didn't find it terribly easy, actually. I had a rather good time last night. So I just want to say, first of all, what an honour it is to be speaking here. Obviously, first of all, on account of my chromosomal handicap, but also more generally, just it's such an honour to be part of the Liebert family. Really, I've edited one of Marianne's journals for 10 years now, almost. And every single moment I've worked with the organisation, from top to bottom, has always been an absolute joy. So I really feel honoured to be involved. Putting it another way, actually, so obviously I'm a heterosexual white male. I don't actually experience very much discrimination and so on. But I do have one example of that because originally I was trained as a computer scientist and I moved sideways into biology. With the result that I've never been trained in how to work a pipette or run a gel or anything like that. And some of my colleagues in gerontology tend to rather look down on me for that and presume that I have nothing useful to say because I didn't do my time, so to speak. So in that sense, some of the things that were being mentioned to do with the patronising ways in which various people here have been treated over the years, they do resonate somewhat. But of course rather less so perhaps than in the case of sexual discrimination. Yes, so today I thought I would actually obviously talk about what SENS Research Foundation does and how how we're able to move things forward in developing therapies that might actually postpone the ill health of old age. Of course, the reason we're able to do it is because far-sighted people have allowed me to obviously not only edit a journal, but also to create this foundation and actually get research done. But there's not nearly enough of this work going on yet, and I hope I'll be able to convince you that it's time for a change in priorities, in research priorities.
I forgot to give a background, your background. Why don't you give a quick, because it's fascinating.
Okay, sure, yes. As I briefly mentioned, I was originally a computer scientist. I grew up in London, went to Cambridge University about 30 years ago as an undergraduate, and I did research in artificial intelligence for about 6 or 7 years after graduating. During that time, I met my wife, who is considerably older than me. She was already a full professor at UC San Diego at that time. This is 1990. And she was on sabbatical in Cambridge. And over the next couple of years, I learnt a lot of biology by accident over the dinner table because I'd given up formal biology education at the age of 15. And I went back to the University of Cambridge in kind of hybrid capacity, working on a bioinformatics project, which I did for the next 10 years and then part-time for another few years. It was an interesting and very fortunate way to be able to essentially switch careers. I kind of discovered by accident that biologists were mostly not very interested in aging and didn't really feel that it was a particularly important thing to work on, which was a complete eye-opener to me and utterly horrific. I had always presumed that biologists all perfectly well understood that aging is the world's worst problem, but apparently this wasn't the case. So eventually I decided I had to do something about it myself, and I was able essentially to create a new career for myself in my spare time, which I had plenty of because this particular bioinformatics project was very undemanding. And yeah, so the rest is history, really. I started editing Rejuvenation research in 2004 when the previous editor of what was previously called the Journal of Anti-Aging Medicine decided to step down. And is Vicky here? Is Vicky here this morning? I owe Vicky Kern a lot because when she asked me to take over from Michael Fossel, I said no, and she came back a month later and asked me again, and I said no again, and she came back a third time, and I finally just realised that there was a way in which I could go part-time at my university job and thereby make the time to be able to do the job, and so I got lucky there, as in on many other occasions. All right, so yes. So what do we do? Well, I'm interested in doing something about aging. I'm not in favour of the idea of getting sick just because you were born a long time ago. And so this is what SENS Research Foundation does. I first want to just give you a nice, concise definition of aging. Everyone, of course, has their own intuitive definition, but it's nice to make sure that we're all on the same page in a concrete way. This is the definition that I tend to use. Aging is a side effect of being alive in the first place. It's simply the accumulation of molecular and cellular damage as a variety of different side effects of the body's normal operation. And the body is, of course, set up to tolerate a certain amount of this damage because that's why we don't get particularly unhealthy until middle age or later. But eventually the damage exceeds the threshold that the body is set up to tolerate, and that's when the diseases and disabilities of old age emerge and progress. So the question then is, what in principle could we do about it? And pretty much everything that we have today in the clinic that we might call geriatric medicine is this kind of approach. It's essentially trying to treat the diseases of old age the way we would treat other diseases, like infections and so on, banging away at the symptoms and trying to eliminate them from the body so that the body remains healthy. Now, if we just think about the definition of aging, and the definition, therefore, of the diseases of old age, we can see that that's never going to work. It's always going to be, at very best, a very modestly efficacious approach, simply because the diseases and disabilities of old age are consequences of this accumulating damage, and therefore the geriatrician's job is going to get progressively harder as the patient gets older and this damage accumulates and puts more and more pressure on the progression of the pathologies. So it's better than nothing, but it's not much better than nothing, and it never ever will be. And I am by no means the first person to point this out. It's probably 100 years now, possibly more, that people have been kind of realizing this and saying, well, okay, we'd better adopt the policy that prevention is better than cure and dive in at an earlier stage in the chain of events. And so what I'm calling here the gerontology approach is essentially to clean up metabolism, to try to tweak the way that the body works in such a manner that it creates damage at a slower rate than it normally would, and therefore, of course, to postpone the ill health of old age. Fine so far. Okay, but it's really not looking too good there either. Because just as the pathologies of old age are extremely numerous, you're obviously not expected to read this slide, quite a lot of things go wrong when we get old, as we all know. Similarly, the gerontology approach of cleaning up metabolism is rather challenging for kind of the same reason. This, as all of you know, is a simplified diagram of a small subset of what we know about how the body works. And that's bad enough, but the real problem, of course, is that this is a simplified diagram of a small subset of what we know about how the body works, which, as we're all aware, is completely dwarfed by the astronomical amount that we don't know about how it works, even ignoring all the stuff that we don't even know that we don't know. So the gerontology approach is not doomed in principle the way that the geriatrics approach is, but it virtually is. It's an idea whose time has emphatically not come. We don't understand biology anything like well enough to have the faintest chance of being able to tweak this so that it does something we don't want it to do, namely the creation of damage more slowly than normal, without having unintended side effects that do more harm than good. It's just not going to happen. Plus also, of course, we've got the minor detail that slowing down the accumulation of damage isn't much use for people who are already carrying a lot of it around. People who are already on the point of becoming ill as a result of their age, we're not going to be able to give them very much benefit. It would have to be a kind of lifelong therapy in order to have any significant effect. So it's all looking really rather dismal, isn't it? And indeed, gerontologists, by and large, as of 10, 15 years ago, were pretty unanimous in their view that we shouldn't even bother talking about intervention in aging because it's just so far away. And it's better to act like seismologists. Seismologists understand that the thing that they study is bad for you, but they do not propose to do anything about it. It was terribly, terribly unfashionable to talk about intervention when I started getting into gerontology. However, that's not the end of the story. Coming in sideways into gerontology was very helpful to me because it allowed me to think about the problem from a new perspective without being encumbered by all the conventional wisdom of the field. And this is more or less where I started. I said to myself, well, okay, look, the human body is a machine. It's a really complicated machine, but it's still a machine, and therefore we ought to be able to get some hint as to how to extend the healthy longevity of this machine by examining how we already successfully extend the healthy longevity of simple man-made machines beyond what they were designed for. So here's a car that was not designed to last as long as it has. It was probably designed to last 10 or 15 years before it falls apart, and it's actually lasted maybe 100 years. How do we do it? The answer, of course, is regular, comprehensive, preventative maintenance. We do not adopt the geriatrics approach of waiting until the doors fall off and then putting them back and having them fall off again and so on. We also do not adopt the gerontology approach of somehow, improving the nature of the car so that it doesn't rust so easily, things like that. We just can't do that. We don't know how to, you can't really make a car intrinsically more longevous when it's already been built. But what you can do is preventative maintenance. And that turns out to be a perfectly good analogy. If we go back to the human body, we can think about the possibility of not trying to slow down this process where metabolism creates damage, or this process where damage eventually translates into pathologies, but rather uncoupling those two processes from each other by diving in and periodically repairing these various types of damage, not necessarily completely, but well enough that the overall abundance of damage stays subpathogenic. And I believe that this is a very much more promising approach. It seems to avoid the complexity of metabolism, because we're essentially saying let's leave metabolism alone and have the damage be created at the natural rate. And it also avoids the complexity of pathology, because the nature of damage turns out, as I will shortly describe, to be considerably less complex than the nature of the pathologies that it leads to. In particular, the big kind of breakthrough that led me to feel optimistic about all this back around 2000 was the realization that all of the various types of molecular and cellular damage that gerontologists would talk about, that would be, considered likely to contribute to eventual age-related ill health, could be classified into a very manageable number of categories. And here are those categories. What is cell loss? It's just cells dying and not being automatically replaced by the division and differentiation of other cells. And we know very well that this contributes, indeed it's the dominant contributor, to certain types of pathology of old age. The example that I normally use, because it's so clear-cut, is Parkinson's disease, which is, of course, driven by the loss of dopaminergic neurons in the substantia nigra. And we can go down this list and we get the same kinds of things. So what do I mean by division-obsessed cells? I simply mean cells that divide when they're not supposed to, and therefore you get too many of them. Of course, that's more or less the definition of cancer. Death-resistant cells, having too many cells of a particular type because they're not dying when they're supposed to, they're not responding to apoptotic signals. This turns out to be really important in the aging of the immune system, among other things. Mutations in the mitochondrial DNA. This is actually a rather controversial one still. People have been worried about mitochondrial mutations since forever, but there has been a lot of difficulty in determining precisely how the accumulation of mitochondrial mutations contributes to age-related ill health. There's plenty of circumstantial evidence that says it does, but the details are still unclear. That's good enough for me, though. I figure, we shouldn't be continuing to focus on discovering whether something matters or not. If we've established that it's hard to answer that question, then it's best to just get on and try and deal with the problem. And if we end up finding out that we've solved a problem we didn't need to solve, then no harm done. Whereas if we decide let's worry about it later, and then we develop everything else, and the mice and then the humans carry on dying on schedule, we're going to feel pretty stupid that we didn't fix this. Molecular garbage, so simply molecular byproducts of normal metabolic processes that, for whatever reason, the cell does not have the machinery either to excrete or to break down, and so they accumulate. Mostly this happens in the lysosome, of course. I'll be coming on to an example or two of that later on. We get, of course, amyloid as well, and other material accumulating in the extracellular space, the most famous amyloid being, of course, senile plaques in Alzheimer's disease, but there are other examples in other tissues that turn out to be quite important in various aspects of aging. And finally, the stiffening of the extracellular matrix, the accumulation of random molecular crosslinks between proteins that compose the extracellular matrix. This turns out to be the main driver of arteriosclerosis, among other things, and it is the consequence of reactions between certain amino acids of extracellular matrix proteins with circulating sugar molecules, or at least that's the overwhelming majority of the source of this stiffening. So, a couple of questions about this classification. First question is, how can I be so confident that it really is an exhaustive classification, that there is no eighth category? Well, one can make a biological argument. One can say, what are we made of? We're made of cells and stuff between cells. What are cells made of? And so on. And then one can use the principle that damage can only, in principle, accumulate in long-lived structures. Because, if it's a cell, or for that matter a molecule, is damaged and then broken down, then the damage is gone. So it's not accumulating. So it can't contribute in its own right to the eventual pathology of old age. And so one can more or less get this list that way. But there are also rather persuasive circumstantial arguments that this is a comprehensive list. I like this one, the fact that it's been the same list for more than 30 years. All of these things have been major topics of discussion and research within gerontology since at least the early 1980s. And you would kind of think that with all that we've discovered and so on about how to analyze biological systems in that time, that we would have added to this list if there were any adding to do. Now, a weakness of that argument is, well, yes, they've all been discussed, but no one was actually challenging the community to come up with a comprehensive classification of aging. And that's true, but I have been, and I've been doing it for more than a decade now, and it seems to be standing the test of time. Really, it's an important question, and so I have been asking it, and I seem to be getting away with this classification. The other thing I want to mention is, why is this classification useful? I mean, obviously there are lots and lots of different types of damage that accumulate, and you can categorize 1,000 things into 7 different categories in a lot of different ways. So what's so good about this particular way? The answer is the type of interventions that are involved. The claim I'm going to make, and try to justify shortly, is that within each of these categories, each of the examples can be addressed by more or less the same type of therapy. So the details are different from one example to another within a category, but only the details. So if we look at, for example, cell loss, the typical way to repair that, to implement the maintenance approach that I'm calling it, is, of course, stem cell therapy. The cells aren't being replaced automatically. We put in progenitor cells of some kind and allow them to proliferate and differentiate to replace the cells that are needed. And, of course, stem cell therapy is different for different tissues, and that's why stem cell research is such an enormous field. There's a lot of different little specializations within it that need to be addressed. But the reason it's a field at all is because there's a great deal in common between different stem cell therapies. And so, so this is the kind of principle. And the same thing applies across all of these 7 categories. All right, so I want to spend a little time emphasizing the relationship between aging, as defined in this way, the accumulation of these various types of damage, and the diseases of old age. And this is something that I'm probably going to skip through pretty quickly because from the perspective of biologists, it's pretty straightforward, but it's something that I think is very much worth emphasizing, and I emphasize it at greater length generally when I'm speaking to non-specialist audiences. Sometimes the relationship is very straightforward. As I mentioned earlier, this concept of cells dividing when they're not supposed to is basically the definition of cancer. So it's just a simple one-to-one relationship. But if we look at heart disease, for example, there's lots of different types of heart disease that happen to the elderly, and they come under different categories. So if we think of, for example, atherosclerosis, that's ultimately driven by the accumulation of oxidized cholesterol in the lysosomes of macrophages in the artery wall, which poisons them, and eventually they become foam cells, these kind of undead inflammatory things that accumulate and become fatty streaks and plaques. And I'll be talking about that a bit later, but that comes under this category, intracellular junk. It becomes extracellular when the macrophage disintegrates and becomes the necrotic core of the plaque, but we don't need to worry about that yet. We've also got, however, other types of heart disease. We've got, as I mentioned a moment ago, arteriosclerosis, stiffening of the arteries, which is basically number 7 down here, the accumulation of crosslinks that result in less elasticity of the major arteries, results in higher blood pressure, with all the consequences that has for other organs, of course. Then there's the problem of amyloidosis in the heart. Recent discovery over the past 5 or 10 years has been that extremely elderly people, people 100, 110, very, very often die of senile cardiac amyloidosis, the accumulation of something very like the senile plaques you see in Alzheimer's, but whose major component is a different protein. And this stuff, basically just gets in the way and mucks up the way the heart can work. The reason it was only a recent discovery is fundamentally because centenarians are very rarely autopsied, and so there just wasn't enough data. But that's been done a little bit now. Finally, there's people simply, their heart stops for no other particular reason than that there is no sufficiently strong signaling between the brain and the heart to keep the heartbeat going. And that ultimately arises from the loss of cells, of pacemaker cells in the sinus node. So this is this category up here. So you can see heart disease, of course, there's no one heart disease in aging, and all of these things can happen. Alzheimer's is actually something I want to say a little bit more about because it's a great example of a disease which is defined as a single disease, but it's defined in terms of a multiplicity of phenotypes. So, of course, the actual original definition from LS Alzheimer back in 1908, I think it was the combination of these two things, senile plaques in the extracellular space in the brain, and, of course, tangles in the neurons themselves. And, of course, we're also aware that as Alzheimer's progresses, there is cell loss spreading from the hippocampus to other areas. So all of these three things are characterized by Alzheimer's. And, so it's a many-to-one relationship, but it's still a clear relationship. If we look at the things that we don't give disease-like names to, then they're often lumped together under terms like frailty or death from natural causes or whatever. They are ultimately pretty much everything here, except you could argue not cancer, is degenerative in a kind of disparate, diffuse kind of way. And they all go together to cause the loss of cognitive and, of course, physical function that is seen in old age. So as we can see, the relationship between aging and the diseases of old age, or indeed the disabilities of old age, is very clear. And indeed, you don't really need to be a biologist to know that the diseases and disabilities of old age must be parts of aging. How else could a disease or a disability of old age be old age specific? How could it be predominantly affecting people who were born a long time ago, other than by virtue of being some aspect of the later stages of a process that goes on throughout life? It's obvious, really, but apparently it's not obvious to gerontologists, so I've been having to repeat this quite a lot. All right, so now let's get on to what we're actually going to do about it, because, of course, classifying, dividing the problem into bite-sized sub-problems is a great start, but we've got to figure out how to actually address each of these types of damage. And ultimately, it comes down to four types of approach that begin with R. Sometimes we can do replacement, sometimes removal, sometimes repair, sometimes reinforcement. And if we go back to the 7 categories that are here, then this is how it breaks down. I've already mentioned stem cell therapy as essentially what it's all about in implementing the maintenance approach for cell loss. That's obviously replacement. Getting rid of molecular garbage, whether inside or outside the cell, is obviously just removing the stuff and allowing the cell to continue to function. And I'll talk a little more about that in a moment. Also, as I mentioned, we have this concept of cells which become blind to apoptotic signals. The immune system I mentioned earlier accumulates especially cytotoxic T cells of a state that's called anergic. In other words, essentially they still produce cytokines, but they no longer proliferate in response to their cognate antigen. And these accumulate to really impressive levels in the elderly, generally as a result of the presence of persistent infections such as the herpes virus family. Cytomegalovirus turns out to be public enemy number 1 in this. Seems that when it reactivates and, of course, thereby stimulates proliferation of memory T cells that have hung around since the last time the CMV activated, then eventually the memory T cells just won't proliferate anymore. But the thing is, unfortunately, they also don't die. Of course, the standard way the immune system is supposed to work is that after an infection has been eliminated, almost all the cells that proliferated to do the elimination go away, and you're left with this very small residue of memory cells to respond to the next time the infection comes back. This no longer happens. And what people often call the immunological space is increasingly occupied by these unwanted cells, with the result that proliferation of other T cells to fight other infections is inhibited. And so we'd like to get rid of those cells, and we're working on ways to do that. The two cases which I talk about here as reinforcement, well, I've only got 45 minutes. Thank you very much to Carla for giving me a little extra time for this talk, but I don't have really enough time to go into this to do these justice, so we can talk about them later. So what do we do? Well, SENS Research Foundation is quite small. Our budget last year was less than $5 million, and we fund as much as we can. We have 3 major projects in our headquarters in Mountain View, and just over a dozen projects in universities around the world, mostly in the US. And we certainly try to cover all the bases, but we set our priorities mainly on the basis of what other people are doing. In other words, how we can make the best bang for the buck, so to speak. And that means we don't do much in category number 1 in relation to stem cell therapy, simply because it's a big field already. There's lots of people out there doing it. Whereas the other ones down here we do a lot more. The ones with 2 exclamation marks next to them are the ones that we feel are particularly neglected by the research community, and therefore we're putting the most effort into them. I'm going to talk about this one just as a kind of case study a little bit more, because it's probably our biggest actual breakthrough so far, and it's going quite well, and also it's really rather important. I'm going to talk specifically about the molecular garbage problem in relation to atherosclerosis. So we have a project in our lab in Mountain View looking at the same kind of problem in relation to macular degeneration, but the work on atherosclerosis, which we're supporting at Rice University, is a little bit further along. There's a little bit more to say. So as I mentioned earlier, atherosclerosis begins with the accumulation of foam cells, which are undead macrophages that have penetrated the artery wall, and they've become undead by virtue of being essentially poisoned by contaminants in the detritus that they are going there to clean up. The detritus is predominantly lipoprotein particles made of cholesterol, cholesterol esters, and such like, and macrophages can process that kind of stuff no problem. The difficulty is that a small proportion of that material has become spontaneously oxidized into forms that the macrophage does not know how to handle, and that's what we need to deal with. So, how do we deal with it? Well, good. So it turns out that the molecular nature of these contaminants has been well characterized over the years. And the main problem is one particular type of oxidized cholesterol derivative called 7-ketocholesterol, 7-KC. So that's what we've been going after for the past several years now. And our first step was to look for bacteria, or indeed any other species for that matter, but bacteria are easiest, that are able to break down 7-ketocholesterol. And it turns out that it's not very difficult to find such bacteria. We used the same methods that, that are used in environmental decontamination, in bioremediation. We just did what's called an enrichment culture. We got a whole bunch of bacteria from different environments, gave them 7-ketocholesterol to metabolize, didn't give them anything else to metabolize. So they can't grow unless they know how to break down this stuff. And it worked. We've got a whole bunch of strains here that couldn't do bugger all with this stuff, and they're just sitting there like lemons. But these 2 strains were perfectly happy, and after only 10 days, the material's entirely gone. So that was very nice. Now, of course, the idea here, the next step, is not to do what they do in bioremediation and simply, introduce a large number of the relevant bacteria into the relevant environment and make the stuff go away. Chances are, if we injected these bacteria into the bloodstream, there would be side effects, really. The idea is to figure out the genetic basis for what they do. In other words, what genes and enzymes they have that allow them to break down 7-ketocholesterol. Woo! What happened there? Bullshit. Let me try that again. It'll be all right. So we took a couple of different approaches. We did straightforward differential gene expression analysis, finding out which genes are upregulated when we give them when we give the bacteria 7-KC, but not when we give them some similar molecule that we don't want to break down. So in this case, native cholesterol. That worked reasonably well. We also tried, as you can see here, simply mass spec. We wanted to find out what the metabolites were, the breakdown products that these bacteria generated in the process of breaking 7-KC down. That was also very, very effective. Long and short of it was, we found an enzyme or two or three that did the job. That's the easy part. We got there a good few years ago. Hard part was to get these enzymes working in a human cell, because of course we've got a bunch to do. First of all, the 7-ketoglycerol is accumulating in the lysosome. Bacteria don't have lysosomes. You've got to do a bunch of stuff to make sure that the enzyme is appropriately targeted. You've got to make it work at a different pH, lots of things like that. But anyway, we got it to work. This just shows that we were able to co-localize. This is a lysosomal stain. This is our engineered gene, and there's reasonable co-localization. And we had to, as I say, mess around a good deal more to make the thing actually effective, but this was what we were able to publish last year. We were able to show that essentially for appropriately chosen concentrations of this toxic cholesterol derivative, the viability, as measured on the y-axis, of cells which have our enzyme suitably targeted to go to the lysosome is much better than these various negative controls, which are like cells with no enzyme, or cells with the wrong enzyme, or cells with the enzyme that's not targeted. If you've got an absolutely insupportable amount of this stuff, then all the cells die anyway. That's just kind of what you would expect. But the recovery is very significant. So we're extremely happy about this. For the past year, we've been trying to reproduce this result in macrophages, which is the cells that really need it. This data is from fibroblasts. And that's turned out to be a good deal harder than we were hoping, but we're almost there. So I'm still very optimistic about this project. Of course, there's a long way to go. Let's not be over-optimistic. The next step is, of course, to move to mouse models of cardiovascular disease and then to clinical trials. And we know it's going to be a while before we really get there. But let's look at the outcome in the end and how worth it this is. We all know what we have today in the clinic for cardiovascular disease, and we also know, of course, that cardiovascular disease is the #1 killer in the Western world, right? So what do we have? We have stents. They're not terribly useful, really. They're not terribly effective. We have statins, and we all know the side effects that are involved there. The fact is, reducing the amount of cholesterol that our bodies make is probably not going to be terribly good for us, given that cholesterol is a rather important molecule. So this is going to attack the problem where it hurts, the real Achilles heel of the problem, and it's going to be a vastly more effective treatment for atherosclerosis than anything that exists today. All right, so I just want to very briefly summarize the other stuff that we do at SENS Research Foundation. We have, as I mentioned, 3 projects in-house. We do a bunch of other projects around the world. I did want to mention our educational program. We have a thriving and rapidly growing program, which involves internships both in our own lab and elsewhere. We also we're in the process of developing online coursework, so we regard this part of our activities as quite important. We've been able to gain a good deal of credibility. I mentioned earlier on that this whole approach is really, I really came in from left field, not just in terms of my own career, but also in terms of the nature of the ideas that I put forward. It was a really big departure from mainstream gerontological thinking, and it did take quite a while for people to take this whole thing seriously. But now we're in a very nice position. All of these people, of course, are extremely world-leading luminaries in their various fields. There are a number of faces here that those at Liebert might recognize because they're associated with various journals. But yes, we're extremely happy to have this kind of level of endorsement. And if you want to know more about the foundation, you can go to our website and download the annual report or the research report, which is the one that has most of the detail about our projects. And I wrote a book a few years ago which covers all of this in excruciating detail. I call it semi-technical because I wrote it in such a way as to allow non-biologists to read it. It doesn't rely on all that much jargon, but fact is, even an educated scientist who's not a biologist will have to go pretty slowly in order to go through this. And it does not cut any corners in terms of the detail, that's for sure. All right, so that's all I want to say about the biology. I'm now going to move on to the kind of the social context of this and really, how this fits into the theme of this meeting and how one really ought to think about this whole thing. And I do this for a couple of reasons. I spend at least as much of my time doing outreach work one way or another, on camera or on stage, talking to non-specialist audiences about not only the feasibility of postponing age-related ill health, but also the desirability, which turns out to be something that a lot of people have doubts about. Now, we must ask ourselves, first of all, why on earth would people have doubts about that? And ultimately, it comes down to what I'm describing on this slide. I haven't yet told you anything about how effective I think these therapies are going to be. So let me actually put a number on that. I think that the approaches that I've outlined to you today of repairing the various types of molecular and cellular damage that constitute aging have a good chance of being developed within the next 25, 20 years, something like that, to a point where we can take middle-aged people, let's say for sake of argument people aged 60, and give them 30 additional years of extra life. So in other words, these therapies will not be 100% perfect, they won't be able to absolutely eliminate all types of all damage, but they will be close enough to that goal that we will be able to rejuvenate people who are 60, both mentally and physically, of course, sufficiently well that they won't be biologically 60 again until they're chronologically 90. That's the definition of where I think we can get within the next 20 or 25 years. And that is, it sounds wonderful, and it's a good deal more impressive than anyone is really willing to claim for any other approach to combating the diseases and disabilities of old age. But arguably, I made a mistake back in, a decade or more ago now, when I started talking about this. I told people what I felt about the longer term, about what would happen next. And long and short of it is, I think that what's going to happen next is much more dramatic. If we look at the mortality rates that we see in today's Western world, we can see that, if you get to the age of, let's say, 26, your chance of not reaching the age of 27 is less than 1 in 1,000. That's where the mortality rates are. So that means the only reason we live as little time as we do is because of aging, because that number goes up with chronological age. In fact, it goes up exponentially with a half-life of something like 7.5 years, a doubling time. That if it didn't rise with age, obviously, by definition, most people would live to have 4-digit lifespans. And that is just with today's risk of death from causes that do affect young adults, like car accidents or whatever, which, of course, are quite likely to come down. So, in fact, this is a conservative estimate of average lifespan in the absence of aging. So the question is, so what? I mean, as I mentioned earlier, I'm only expecting these therapies to be imperfect and to give 30 or so years of extra life. But the thing is that simply by the very nature of being rejuvenation therapies, rather than therapies that just slow aging down, these therapies buy time. If you're 60 years old and you get fixed up well enough that you're not going to be biologically 60 again for another 30 years, that's 30 years of time to do more research, to improve the comprehensiveness of the therapies, so that when you're chronologically 90, you can be re-rejuvenated, so that you're not biologically 60 again until you're chronologically, 150. Now, you can see the point, right? You can carry on doing this. All we need to do is stay one step ahead of the problem. And I've called this longevity escape velocity, essentially the rate at which we need to improve the comprehensiveness of the therapies in order to allow a situation in terms of people's actual health for their age that is identical to how it would be, to how it would be if we actually had 100% perfect damage repair therapies. Even though we never do have perfect therapies, it would be equivalent. Now, I said that it was maybe a mistake for me to start talking about this a decade or so ago. And the reason I say that is because it got me into a humongous amount of trouble. It was the thing that really gave me a reputation as a heretic within gerontology. Because my mainstream colleagues, especially my more politically sensitive mainstream colleagues, said to themselves two things. Number one, this isn't science. You're not telling us how we're going to do these re-rejuvenations, these gradual improvements, therefore we don't believe it's going to happen. Now I say, correct, it's not science. It's extrapolation from what type of therapies these are, and from what little we know about how those therapies might in the future be refined. But to say we don't know whether this is going to happen, therefore I choose to believe it's not going to happen, is also not science, right? It's irresponsible to say that something's not going to happen because we can't see how it's going to happen. On the basis of what we know, and I completely grant that we don't know very much, on the basis of what we know, it is actually vanishingly unlikely that we would fail to achieve and maintain longevity escape velocity once we had got this amount of first-generation rejuvenation, so to speak. So, you know, I've had a bit of a running battle in my field on this point. I think it's absolutely essential that those of us who understand the feasibility of these first therapies are also willing to actually tell it like it is with regard to what that might mean in the longer term. The other reason, of course, why my mainstream colleagues were a little bit unhappy with my predictions is that they are so dramatic. And, you know, this gets terribly good headlines and everything, but some people get worried that it sounds like science fiction and therefore it will bring the field into disrepute. And again, I think, you know, that's an absolutely irresponsible way of looking at it. If we simply say things that we don't believe in order to curry favour with people who are politically sensitive, then progress doesn't happen. So I'm not in favour of going about things that way, and I feel especially that someone like myself, who is not in a standard tenured or pre-tenure, it would be even worse, of course, academic position, someone like me who can get away with saying things like that has the particular responsibility to go and do so. So, what does this mean? Well, speaking to an audience that's mainly women is interesting for me in this regard, because I have found over the years that the negative reaction to the sorts of things I'm saying here tends to be stronger from women than from men. And I'll give you a few examples. These are the sorts of things that I have to discuss all the time with journalists and so on. People say, oh dear, if we don't have aging anymore and everyone carries on, everyone lives to thousands of years old, then we're not going to have anywhere to put people. And they'll say, you know, this is only going to be for the wealthy, you know, it's going to increase inequality of access. The other amusing thing is that people say both of these things together. You know, it's pretty bleeding obvious that they are mutually exclusive. Either, you know, right? They say this, you say, oh dear, you know, dictators are going to live forever. You know, last time I looked, dictator came fairly high on the league table of risky jobs. Not many people, not many dictators die of aging in the first place. People say, oh, you'd be so boring, you know. Dear me. And of course, this question of how would we pay the pensions, you know, if people retire. Yeah. So I have to address these problems all the time. I'm not going to spend much time on them now, not least because Carla has reminded me that I don't have very long. But the fact is, I don't think much of any of these problems. I have very good answers explaining why this shouldn't be something we worry about. This is what we should be thinking about when we consider the sociological implications of doing something about aging. We won't have to cope with the astronomical burden of age-related ill health. As you all know, 90% of the medical budget in the Western world is spent on diseases and disabilities of old age. Not only will we not be doing that, we will be paying for these therapies. They will be paying for themselves because the elderly will continue to be contributing wealth to society rather than just consuming wealth. As regards boredom, well, I mean, you know, as far as I'm concerned, a lot of people who haven't been properly educated are pretty bored today. You know, they can't think of anything to do but watch television. But I personally, having had a good education, have at least 1,000 years of backlog of things I haven't had time to do, and by the time I'm through it, I'm sure I'll have another 10,000. So, of course, this is a problem of education, which is something that we could spend some of the money we're saving from not having to keep people alive in a bad state of health. Flexible career structures, of course. So this is something that's, of course, very relevant to a number of the discussions that we had yesterday. One thing that we're not going to have anymore in a world where aging has been eliminated by regenerative medicine, effectively, is menopause. Menopause is something that ultimately arises from the aging of one particular organ and creating either new ovaries by tissue engineering or rejuvenating the ovary using stem cell therapy or, you know, a variation on those themes is something that is no more intractable than the corresponding therapies for other organs. So it's my belief that we are going to be in a position of allowing women to have kids safely at any chronological age, and the consequences of this for one's career choices are, of course, enormous. one doesn't really need to think in terms of a biological clock anymore. I hardly need to go into what this amounts to, but I will mention that, of course, it's not just the choices that the woman makes that are broadened, but also the anticipation of those choices that will be made by employers and deans and people who will be able to take into account the let's call it the diminution of the risk that someone will just leave their job and go and have children. That sort of thing will be very different in a world where aging doesn't exist. And of course, a lot of the elderly, the real problem with selling this to the world is that the elderly have this infuriating habit of not complaining about being sick. They seem to actually prefer medical expenditure to be focused mainly on the young, and this is, of course, reflected in public policy. They don't want to be a burden on their kids. And this, of course, also has economic considerations as well. The fact is, one is less productive if one has to look after one's sick parents. So, all of these things are what we ought to be taking into account in terms of the sociological considerations, not over and above, of course, the sheer humanitarian aspect of keeping people healthy is a good thing. And this is why I believe that it's no contest. This is the world's biggest problem. I am extremely privileged to be spearheading the mission to fix it, and that's it. Can we manage a couple of minutes of questions, you think?
You can. One first question. Every action has a reaction, and your approach with the enzymes is fascinating, and you're going to the end products of the enzyme action, but those end products can, in fact, have problems, so you...
Sure, we're very aware that the breakdown products of approaches like this also need to be examined. So sometimes, of course, it's not a problem. So, for example, the ideal, simplest scenario would be an enzyme that simply reduces 7-ketoglycerol back to native cholesterol. We know that native cholesterol is okay, so that would be done. In practice, that's not what we've found so far. The products we've found are other oxysterols, but it turns out that they're less toxic, and so, yes, but obviously it is something that's part of our research program.
Good. Any other questions?
Your first and last sociological implications, delaying the ravages of aging and then burden to children, isn't that just postponed to a later time?
Yes, but it's postponed indefinitely. This is the thing. I mean, so the idea is that these first generation therapies come along and give you an extra 30 years, but by the time that 30 years is up, the therapies have been improved to give you another 30 years, and so on.
So indefinitely.
That's right.
This is very provocative, and I think it's a...
Yeah, that's
it's
one of the more
interesting.
that's
It's
one of the more
interesting.
polite words that have been used about
Yes.
that. Right.
No other questions. I think they're daunted.
Yeah.
So
Well,
let's...
I'll be here until the end of the meeting, by the way. My
Right.
flight isn't until 10 o'clock or something like that. So.